Good morning, friends. Good morning, students. In today's class, I will discuss homopolysaccharides. So, first of all, I will start with definition of polysaccharides. Right? Poly means many, and saccharides means monosaccharide units. Right? So, polysaccharides contain multiple monosaccharide units. Right? Now, coming on to the definition, they consist of repeating unit of monosaccharides. Right? Mono means single, and when there are multiple single units, they become polysaccharides. Right? And what are they useful for? They are storage form of energy. Right? And moreover they have good structural functions as well, right? Polysaccharide exist in both linear and branch chains as well, right? Now coming on to the category of polysaccharides, they are divided in two categories. First is homopolysaccharide and second is heteropolysaccharide. Homo means same and hetero means different, right? So homopolysaccharides, they contain a single monosaccharide unit, and heteropolysaccharides, they contain two or more monosaccharides unit, right? Now coming on to the next homopolysaccharide. So starch, it is a carbohydrate reservoir plant, right? And it is a homopolysaccharide, right? Homo means it consists a single unit. The single unit is D-glucose, right? So it consists of repeating monosaccharide units of D-glucose, right? And they are further divided in two categories. First is amylose and second is amylopectin. Amylose is unbranched and amylopectin is branched, right? And the sources are vegetables, cereals, roots, and tubers, right? Now, I told that it starts divided in two categories, that is amylose and amylopectin. Now, I'll start with amylose first. Now, starch has two components, that is amylose and amylopectin. The amylose is water soluble, that it is soluble in water, and amylopectin, that is water insoluble, it is insoluble in water. But the content of amylose is only 15 to 20%, and amylopectin is 80 to 85%, right? Now, coming on to amylose in details. So it is long and unbranched, right? It doesn't contain any branch and linear polymer of alpha 1414 D glucose units, right? And it has 200 to 1000 D glucose units as well. And the branching is alpha 14 glycosidic bonds, right? Now coming on to amylopectin, it is branched, right? So at branching we have alpha 16 linkage, right? And in straight chain we have alpha 14 linkage as in amylose, right? And it look like say tree, that because it has branches, so it is tree like, right? So when it is hydrolyzed by amylase, that is pancreatic or slavery, it will liberate dextrin first, then maltose, that is disaccharide, and finally glucose, right? And amylase acts specifically on alpha 14 glycosidic bonds, right? Now here comes the structure. So this is amylose and this is amylopectin, right? In amylose, I told that there are no branches, right? So it is straight chain and alpha 14. So this is one glucose unit, this is second glucose unit, and this bonding is alpha 14 glycosidic bond. So we have this amylose, right? Now in case of amylopectin, the molecule is branched, right? So this is straight chain, and branch is at alpha 16, right? So how to draw branch from 1 to 6 carbon? This is branch, so it becomes this linear chain amylose. Linear chain amylose, when branch comes, it becomes amylopectin, right? So amylose is straight chain alpha 14 linkage only. Amylopectin straight chain with branch at alpha 16, right? Now coming on to the next, that is dextrins, right? They are the breakdown products of starch, right? And starch can be hydrolyzed to first dextrins, then to disaccharide maltose, and finally glucose, right? Now coming on to dextrins, they are glucose polymers produced by microbes, right? So when glucose polymers are produced by microbes, they are known as dextrins, and they are useful where as plasma volume expanders in transfusion, right? And they are also used in gel filtration chromatography, right? Now coming on to the next, our homopolysaccharide, that is glycogen. Starch is plant product, and the animal starch is known as glycogen, right? It is found in humans, right? And it is carbohydrates of animals, right? And location high concentration liver, muscle, and brain, right? And it is also found in non-chlorophyll containing plants as well, right? And it is also found in yeast and fungi as well. And the branching is same as in starch, 1,4 and 1,6, 1,4 in straight chain and 1,6 in branching chain, right? So number of glucose unit depend on the source of glycogen, right? Now coming on to the next molecule, that is our cellulose. The starch and glycogen, they are basically similar in that they have same branching, alpha 14 in straight chain and 16 in branch chain. But starch is found in plants and glycogen is found in animals. But this molecule cellulose, is the difference is that it has bonding. The normal is alpha 1 4, here the bonding is beta 1 4, right? So our amylase, both salivary and pancreatic amylase, they cannot adjust this beta 1 4 linkage. So the molecule cellulose is not adjusted by humans, right? So it is also found in plants and constant of plant cell wall, and it cannot be adjusted by mammals because of the bonding is beta 1 4, and our salivary amylase and pancreatic amylase can adjust alpha 1 4 linkage, so it is not adjusted by the body. So it, what is the function then? It adds bulk to the food and bulk to the faces and helps in decreasing the absorption of cholesterol from the intestine. So more the fiber you eat, the cholesterol level will decrease, right? And now coming on to the our structure of cellulose. In starch and glycogen, the bonding is alpha 1 4. In cellulose, we have beta 1 4, right? So these are two glucose units, beta D glucose and beta D, and the branching is beta 1 4, right? So there comes the structure of cellulose. Now coming on to our next molecule, that is inulin, right? So the molecule of the polymers of glucose was three. First was starch, then glycogen, and then cellulose. Now the polymer or polysaccharide formed from fructose is our inulin, right? And it is water soluble, right? And its molecular weight of around 5,000, right? And where it is found in nature, dahlia bulbs, garlic, and onions, right? So salad products like and the onions, they contain inulin, right? And what's next come the use? It is useful in assessing the kidney function test for GFR measurement. So during GFR measurement, we use inulin, right? Now next comes the molecule, that is our chitin, and it is structured polysaccharide, and it contains repeating unit of N-acetyl D-glucosamine, right? And the linkage is beta 1 for glycosidic bonds, right? So it is found where? It is found in exoskeleton of invertebrates as well as insects and crustaceans, right? So that finishes with our chitin. Now coming on to our so there is the bibliography from where the lecture has been prepared: Harper's, Satyanarayana, Vasudevan, Lippincott, and Chatterjee, right? So that finishes with our lecture on homopolysaccharides. For next video, please like, subscribe, and share. Thank you. God bless. Bye.